Yeah, so I'm guessing there's a range of folks on the call today. I'm guessing some of you have been using Galaxy for many years, but I'm guessing there's also a bunch of new folks here where maybe you've heard about it, or maybe you saw enough in the email that at least we caught your interest a little bit. So, but that's great. We're always excited to kind of reach out to new communities and new folks. So Galaxy is it's many things, so it's a little bit hard to describe in words. Sort of operationally, it's a web-based platform where you can go in and do really sophisticated scientific analysis. So an analogy that I'll sometimes use is on my laptop here in front of me. I happen to be on a Mac. It's a whole operating system where, everything I see on my Mac in terms of the applications and file management and user management is all happening through that operating system. So when I think about Galaxy, I think about it being like an operating system for science where you can have a platform that can support many users, where you can run a lot of different tools, you can keep track of a lot of different data sets, you can run giant analysis over thousands of mammalian genomes, you can do smaller analysis of one gene and one microbial genome. It's very sort of scalable, elastic in that setting there. One really special part of it is for most users, you're going to access it entirely through your web browser. So, be it a Chrome or Firefox or whatnot, entirely through your web browser without any sort of programming experience, you can do really, really sophisticated analysis. You can run state-of-the-art, I don't know, genome assemblers, aligners. You can look for genes. You can look for proteins. You can look at evolution. You can do machine learning. You can look at, all these environmental factors, chemistry, proteomics, and beyond. There's just a lot of really sophisticated capability that's all really embedded inside of your web browser. Behind the scenes, there's a lot of software and computing systems, and we'll say a little bit more about that in a minute. Behind the scenes, there's a lot of sophisticated processing taking place where it's exactly the same tooling, it's exactly the same sort of environment. If you've ever heard of like the Google Cloud, it's sort of that level of sophistication, but it's all accessed through your web browser. And then another really sophisticated part is that it's all for free. You don't need a credit card. You don't need to purchase anything. It's all for free. You can just sort of go to this website and then sign in. It takes 2 minutes. And then you get access to all this great scientific resource, all this scientific capability, all for free. So there's questions about, p-values and t-tests. You betcha. Any sort of standard statistical analysis like that, you betcha. We have a very rich support for that. There's more than 10,000 tools available. There's, I don't know, thousands of workflows available. There's all kinds of sophisticated processing that can take place. There is way if you need to do, custom stats in our notebook type of thing, you betcha. We can also support that. There's a lot of depth there. Realistically, today we're just going to go through like, I don't know, 1% of Galaxy, give you a little taste, but there's a lot of sophisticated processing on the back end. To give a little bit more information, right? So we like to think that, a big draw for Galaxy is the rich set of tools that are available. If you go to usegalaxy.org today, there's something like 10,000 different tools that are available. At a technical level, this gets bundled up through something called a container or a biocontainer, where we have basically every major bioinformatics tool ever published to a first approximation. This is a moving target because every day there's a new paper, there's a new tool, but we work really closely with many major communities to sort of integrate those as quickly as we can. So it's a very sort of dynamic, growing environment where we have all this tooling available to do all kinds of analysis. So I think tools is a big draw. Another big draw is we already have it loaded with many thousands of genomes. So, on the left there, just sort of scrolling through, we have fruit fly and frogs and bacteria and humans. We already have all that reference genomes available. So if you're doing anything in genomics, we have a lot of capability there. I should say also beyond genomics, we also have a lot of reference data available for other sort of scientific disciplines, but given the context of today, we're going to be speaking mostly about the genomics data. So we have tons of data. We have tons of tools. Like I mentioned, if you need to drop in and do interactive analysis in Jupyter Notebooks or RStudio, yep, you can do that entirely through your web browser, get access to that as well. If there's like specialized software you need to run, a big initiative Galaxy recently is to work with something called the Vertebrate Genomes Project to look at genomes from many like mammals and birds and reptiles. It just requires a lot of computing power. Takes like more than a terabyte of RAM, but no problem. We can get you access to a terabyte of RAM. If you want to like take your protein sequences and then fold them up using AlphaFold, no problem. We support that, and that uses GPU support. Again, all entirely for free. So we have a lot of diverse sort of computational resources available to our users. Another cool thing is, as you're doing analysis, it keeps track along the way. So it'll be like on the fly, build up a lab notebook for yourself. So it's like, and if you're doing analysis like me, there'll be times where I'm like really focused and I'm, keeping track of what I'm doing. Oh, but then I have to do something and I come back to it a day later, a week later, a year later. All of that information about exactly what was run and how it was run is all recorded for you in something called a history, which is like a living lab notebook that exactly documents all the data that was used, all the tools that were used, all the parameters, all the versions. So you know exactly where all of your analysis came from. And then the last part at the bottom is it's very much a living system. We work very closely with our very large community of users. And we're constantly bringing in new tools to support new technologies. These days there's a lot of excitement around single cell technology, single molecule sequencing, spatial and imaging data to look at sort of expression, across different organs, across different tissues. And you betcha, we have a lot of support for all of that. It's very sort of dynamic in nature. So a lot of the analysis often starts sort of, one step at a time. It's like you get a new data set. Oh, inevitably you want to kind of get a little QC of it. Then maybe you want to do some first analysis, second analysis, third analysis. A lot of that can be done interactively where you're going to be picking and choose. I want to run this tool, get a result. Then I want to run this tool, get a result. Then I want to run this tool, get a result. Of course you can do that. And in fact, that's how we're going to get started today. But another very powerful thing is once you've kind of worked out the analysis you want to do, it's like, okay, I want to do QC. I want to do some alignment. I don't know, variant calling. I want to like prioritize the variants in some way. Once you've worked out the set of tools, you can build what we call a workflow, where once you've kind of worked it out with one data set and then you want to apply it to 10 more, 100 more, 1,000 more, you can then apply that workflow, that same exact set of tools to any number of additional data sets. So that's a great way where you can sort of get started. You can prototype on one or a few, but then once you're happy with how it's looking, rapidly scale it out to thousands of other data sets. So that there's a lot of sophisticated support inside of Galaxy for sort of designing workflows, running them, monitoring them, integrating on them. There's a lot of support there to support that scientific analyses. And another sort of feature is, yes, you can build workflows on your own, but we also have a growing catalog of like really polished, high quality workflows, something called the IWC, the Intergalactic Workflow Commission, really sort of carefully builds and studies and validates these workflows so that you don't have to get started on your own. Here's just sort of the splash screen. If you want to, do annotation of bacterial genomes, we got that. It's, many stages, of many different tools all put together, but in a few clicks, you can run a workflow with your own genome, get annotation. You want to do quality control, you can do that very quickly. If you have a special data called Hi-C that looks at how the genome is folding up, and it takes a lot, again, it takes a lot of steps to run it, no problem. We have a workflow that will help you with that. And in a few clicks, you can run that workflow and get really sophisticated analysis very rapidly. So in terms of the different application areas, there's many. So here's just a handful that I picked that I thought might be kind of relevant to what we're talking about today. A few of the key citations here. As I mentioned, we're doing this big initiative into vertebrate genomics, where our long-term plan is to develop a reference genome for every vertebrate species on the planet. There's more than 70,000 of these, but we're making a lot of great progress there. We had a major paper last year describing that. Another sort of major area relevant to today's discussion is on metagenomics, where we have thousands of tools, hundreds of workflows available to be able to go from kind of raw data and do a whole variety of different analysis in terms of like which genomes are there, what species are there, which genes are there, do they have any kind of important pathways for diseases or other compounds that are being produced. We have a very rich environment for all that analysis. That'll be kind of the main focus for today. On the human genomics side, there's a big initiative to look and support analysis for human genetics and for cancer genomics in particular. And we've had a number of big papers come out there, kind of being able to work with cancer data of different forms. As I mentioned earlier, there's been a lot of work looking at single cell data where all in your browser, you can load in kind of raw or processed data for single cells and do different analysis visualization across thousands, maybe even millions of cells entirely through your browser. There's been a big effort in proteomics. If you want to do a deep dive on what are the proteins, what are they doing, how are they interacting, how are they, kind of related to sort of different phenotypes or different products in themselves. We have a lot of analysis going back for more than a decade that is available there. And then the final dimension I want to highlight is if you want to do state-of-the-art machine learning, you want to design a convolutional neural network or a transformer architecture, train it, evaluate it, fine-tune a foundation model. Yep, we can do that all within your browser, all for free. So there's just a ton of resources that are available. There's a ton that can be done and much, much more. This is just, I don't know, 0.1% of all the different capabilities, all the different activities that are present there. A couple of metrics. So Galaxy, I'm hosted here from Johns Hopkins, but Galaxy is very much a global enterprise. And we'll say a bit more about this in a second. There's very active Galaxy servers in the United States. There's active work in Europe, in Australia, in China, in South America, in Africa, all over. So basically every major country, every continent has a major sort of Galaxy community going there. And we all kind of coordinate. We all kind of share either software, we share tooling, we share best practices. There's a lot of coordination between them to support each other. So no matter where you are in the world, no matter what time zone you're working in, someone will be there to help you, which makes it really exciting. If it's 2 o'clock in the morning your time, that's the middle of the day for someone else, and we're all there to support each other. So collectively, we have more than 500,000 registered users across all these different deployments. We've had more than 100 million jobs. We have hundreds of millions of data sets, approaching 1 million workflows that have been run, hundreds and hundreds of really high quality tutorials that will walk you through, from soup to nuts of how you get started and how you do the science. And we're also very proud that we've supported more than 20,000 scientific papers that have formally cited Galaxy. The actual true number is many more than this. A lot of people now just say, oh, we use Galaxy for this without a formal citation. But if you go to, say, Google Scholar, it's more than 20,000 already. And it's just growing every day. So it's a really dynamic platform with lots of uses there. So if you've never used Galaxy before, and this will kind of foreshadow the hands-on part later for today, what I'd say is right now, go to usegalaxy.org. It'll take 2 seconds to load it up. In the top right corner, there's a link there that says either login or register. That's what you'll probably see there. So if you've never used it before, click on that login or register link. That'll take you to our start page where, if you have a Galaxy account already, please log in. But if you don't have one already, just go to this link down here where it says register here. Put in your email address, a password, whatever your public name is. I do strongly encourage you to sign up for the mailing list, and then click on create. So in, 3 clicks and a little bit of text there about yourself, you'll be able to create your own account. And that will basically unlock for yourself, your own version of Galaxy where your own data, your own tools, your own history, your own sort of workflows, all of that will be kind of set up for you. That's your way to kind of tap into the power of Galaxy in just, in 3 clicks. So if you've never used Galaxy before, I strongly encourage you, use galaxy.org, top right, log in, register here, and then create your own account for yourself. And that'll be important as we go through the hands-on demonstration so you can actually kind of experience this for yourself. And then, like I said, once you've done that, so here's me logged in as Mike Schatz. Here is an analysis I was running last night of a zebrafinch, and I was trying to look at the genome, and I wanted to see a plot of, this is a plot looking at how the k-mers are present using a tool called GenomeScope that I had run through the sort of toolbox here. So all of my data, all of my workflows, all of my analysis now, kind of organized for me. If I want to, I can choose to share this with other people, but by default, it's all just for me. It's very, kind of private data for me. So you don't have to worry about people stealing it or looking at it. It's up to you to control. If you want to share it with other people, of course, we encourage you to do so. But it's all very sort of private to you as you're getting started. And again, it takes 2 seconds to be able to do this, and is a great way to do analysis. Okay, so that's a little bit of kind of the high level of what is Galaxy, why it's cool. Let me just spend 5 minutes to tell you about how it works, and then we'll pivot into kind of the hands-on exercise. So, when I talk about Galaxy being an operating system for science, there's a lot of software behind the scenes. There's a lot of major systems behind the scenes. I'm not going to go through it in any detail, but the headline being is it's all open source. If you go to github.com/Galaxy project, it's all open source, so you can see exactly how it's working, what it's doing. One thing that is pretty common is people will get started with the public server at usegalaxy.org. But if there's like special tools, or I don't know, there's like, you have access to a lot of computing locally and you just want to sort of tap into that locally. One thing that you might want to consider is setting up your own Galaxy server on your own computers. And sometimes that's convenient if there's like, you want to hook it into your own data center. I don't know, there's special data sets that you want to process, you want to have collected. So if you go to this URL, that's where you can go to get all the code. It's all completely open source. It's all completely for free. And that will help you get set up on your own hardware. And kind of behind the scenes, what's happening is what you see is, a web browser where you can point and click and maybe fill in a few checkboxes. Behind the scenes, though, are exactly the scientific tools that you might run on your own laptop, are exactly the scientific tools that you might run in your own cluster. For example, here is sort of the splash screen for HiSat2. This is a very popular aligner for DNA or RNA reads. And what's being sort of orchestrated is all of this information does go into command line tools. Here we're symbolically linking in files and running HiSat2 build to build an index and setting all the paths and all this stuff. So behind the scenes, there's a lot of command line Unix scientific software being running. But the sort of one upside to it is all of that is sort of tucked away, where if you want to see it, if you need to set variables, if you need to set parameters, you can. But most of the time, you're just going to be in this sort of much more friendly user environment where you can kind of just work through your web browser to work with things. So we're going to turn all this detailed code into a web form. But again, behind the scenes, it's exactly the tools that you may have heard of: HiSat, Bowtie, BWA, SAM tools, Hi-Fi ASM, and on, Kraken. All those tools you may have read papers about, those are the tools that will be executed, but they're going to be executed inside of this Galaxy environment. For our main instance, use galaxy.org. Just 2 seconds. We really partner closely with the Texas Advanced Computing Center down in Austin. They have this giant like warehouse filled with computers, and that's where Galaxy sort of really lives in terms of the hardware. We also partner with other supercomputing centers around the United States, like Indiana University, San Diego, Pittsburgh Supercomputing Center. And then we also have a footprint at Johns Hopkins, where I am. We also work very closely with the team at Penn State. In fact, there's a few people on the call today from Penn State as well. Collectively, this is like, Google Cloud level resources, many petabytes of storage, millions and millions of computing cores available, core hours available throughout the year, a growing number of GPUs, especially for machine learning. For you users, it's all completely free. Of course, nothing is really free. There is a building with computers and IT staff, but for you users, it's all free to kind of just sign in. You don't have to have a credit card or anything like that to use it. Within the United States and Canada, there are a few major instances. That main one is at usegalaxy.org, but we also have other ones for human genetics, for infectious disease, for some of the so-called NIH Common Fund projects. We have a special instance for cancer. We have a special instance. We also work very closely with the team up in Canada to support their researchers. And then globally, as I mentioned, there's all these versions of Galaxy that are distributed around the world, and we work really closely. In fact, we share data, some of the reference data. The human genome is the human genome is the human genome. So we share that behind the scenes to all these different instances around the world, just so that we all get equal access to it. Behind the scenes, right, again, there's a lot of tools, there's a lot of data, there's a lot of users. So there's a very detailed database that keeps track of everything for you. And again, from a user perspective, one of the main outcomes is you get that living lab notebook. You get that history of every tool you've ever run, every data set you've ever looked at. That living lab notebook grows on the fly, but there is a very detailed data model that has millions and millions and millions of records in there. And we'll keep track of that organized for you so that you don't have to. So that's, I think, a huge feature for scientific research, that it's actually reproducible and it's also actually accessible, that all you need is a web browser. So our real goal is to really empower users, really make it democratic and accessible for all. If you need to, if you want to, run things on the command line, you can also work with Galaxy in that way. This is really good. Like, for example, if you have a job that every night at midnight, I need to go fetch all the latest genomes and do a bunch of analysis and crank through it every night at midnight. Sure, we can support that. We have a framework called Planemo that lets you do that. In fact, at the peak of the pandemic, we had this setup where every night at midnight it would collect all of the COVID genomes that had been sequenced in the world, and of which there were many millions, and do a massive analysis every night of how all new variants were emerging and where outbreaks were spreading. So if that's the sort of analysis that you need to do at very large scale, no problem. We can also support that. It's a little bit more advanced. We're not going to go into that today, but I did want to just highlight it often starts in the browser, but if you kind of outgrow what's available there, yes, you can do everything via the command line. You can do everything via APIs to scale it up in an automated way if that's what you need to do. Another great resource for everyone on the call today is training.galaxyproject.org. This is our Galaxy training network, where we have hundreds and hundreds of really detailed and really friendly tutorials. I use these all the time. Especially if I'm trying to do an analysis I've never done before, this is where I go look. They're organized into a few dozen major topics, scientific topics. Just at the top here, we have, climate analysis, computational chemistry, big set of resources on SARS-CoV-2, data science, ecology, and on and on and on and on. There's many dozens of topics there. There's also, if you're sort of new to Galaxy and you want to get, up to speed on how you do different analyses, how you work with different data sets, we have dozens of tutorials on that. So there's lots of resources available to work with you, to kind of support you. So again, if it's 2 a.m., you're not sure what to do, go to the training network here. There's probably a good resource. If it doesn't do exactly what you need to do, it'll get you really close. And then hopefully, once you're in the system and you're really close, in a few extra clicks, you can get where you need to be. For today, a couple of resources I want to highlight. We have a whole set of trainings dedicated to the microbiome. So if you click through there, there's several dozen. Here's one I just picked of them. If you're very interested in, say, antibiotic resistance gene detection, there's a very detailed tutorial there that will kind of work through how can you do quality control, how you can do assembly, how you can look at kind of the overall structure of the genome, and then you can kind of dive right in and look for some of the key genes there. So if today, you're following along, but you maybe missed a couple steps, go check out these trainings that'll have a very detailed click-by-click, step-by-step description of what you need to do. And it'll also tell you why you're doing it. Why do we need to do QC? Why do we need to do assembly? Why are we using this tool versus that tool? It's a really detailed tutorial guide that will get you through all the analysis. And this is available for hundreds of different analysis. So there's probably something very close to what you need to do in there, and it's already available. And then one of the last things I want to highlight is, today's just is your first taste. Just a 2-hour session today. But coming up in just a couple of weeks, May 12th through the 16th, we're having something called the Galaxy Training Academy, which is a global week-long initiative to get users all over the world up to speed on a lot of different training and activities. Last year's event had more than 2,000 people globally. It just becomes really exciting because, no matter where you are in the world, what time zone you're working in, at that scale, you can always sort of check in, you ask other people how it's going. You can kind of get really instantaneous feedback. So if you're interested in Galaxy, but you want to kind of learn more about it, please come check out the Training Academy in a couple of weeks' time. That'll be another great resource. And then the last one I wanted to highlight is, in addition to kind of all these online trainings, throughout the year, we do have a variety of in-person workshops. In fact, I will be in North Carolina in Greensboro. I forget the exact date. Is it June 15th? So that will be my next kind of in-person event in Greensboro. In addition to that, we have a conference coming up in New York at Cold Spring Harbor Labs on Long Island. That's June 23rd to the 26th. It's open to the public. So if you go to jxy.io/gbcc25, that will take you to this page. You can learn more about the agenda. You can learn more about kind of the schedule. This will be a sort of a scientific conference where we're going to be talking about science that is done with Galaxy. There will also be a bunch of tutorials and trainings about how to use Galaxy. So this is a great in-person time to be able to meet people, talk to them, get support, get part of the community. Galaxy is all about community development, all about kind of working together. So, through this event, through the event in Greensboro coming up in June, through other events throughout the year, we would love to meet you in person and work with you in person. There's just so much activity going on. It's just a really exciting time. So let me pause to see if there's any quick questions, and then we're going to transition over to Tyler, who's going to kind of get us up to speed about some of the data we're going to be working with, and then sort of the hands-on activities. Yeah, so I see here in the chat, there's a question about, I enjoy analyzing my data with Galaxy. The interface is simple and easy to use. Yep. Would like to work for Galaxy. Great. Well, we love that. Any interest available for volunteer opportunities? Yes, we are always looking for partners. A great place would be to help out with the Galaxy Training Academy coming up in just a few weeks' time. That would be a sort of a natural entry point. If there's, depending on your availability, your interests. So the Academy is like a 1 week long activity. So, just making yourself available during that week, that would be great. If you actually wanted to kind of really help develop trainings, the right person to talk to is Natalie Whitaker Allen. She's on the call right now. If you drop in your email, maybe you can chat with her. If you wanted to do more than that, I don't know, if you wanted to contribute software, we love that. We do all of our work open source, so you can kind of work through GitHub. You can contribute. If you're interested in a scientific analysis, we have scientific interest groups. Maybe there's a group already out there that you can kind of join in on. Notably, there's a very active microbiome research group in Galaxy. In fact, we had a call earlier today. So if you're interested to kind of work on microbiome analysis in this global community, I can get you connected to that group. We would love to work with you. It's really up to you about what sort of availability you have. If you want to contribute more on the science side, more on the back end side, but there's something for everyone. It's such a great community. There's definitely something for everyone.
We have one more question above Mrs. Kiki Parker. She asks, can Galaxy be used to perform p-value calculations or t-test analyses?
Yeah, absolutely. So, today we're going to be talking mostly in the context of metagenomics. We'll see a little bit of stats when we talk about like species abundance. I think there'll be a little bit about, when we look at like, the sort of p-value, e-value, about protein alignments, that sort of thing. If you're doing like, I don't know, differential expression of genes, yes, that is really supported. If you needed to do kind of a p-value t-test type analysis there, differential expression. We're not going to go through that today, but there's a lot of tutorials, there's a lot of trainings available that would work with you. And then again, in the limit, if there's like, there's a very custom analysis you're trying to do, we support that in many different ways. Through the UI, there's a lot of sort of statistical tools available. In the ultimate, though, if you really need to do custom statistics, that's when you can drop into a Jupyter notebook and do anything that you might imagine through NumPy, SciPy, if that's what you need. But most of the time, there's a tool, there's a workflow that will help you through that without even needing to drop in there. Question here is, I've seen Galaxy EU, I've seen Australia, I've seen Maine. Are those the same platform? Almost entirely, right? We all use exactly the same code base. They may look a little bit different because we let people customize it. They'll be running on different hardware. They might have access to certain data sets that are a little bit different. But the look and feel will be very, very similar. But because they are in different countries and there might be different sort of focuses in different countries, there might be slightly different tools or data, but the look and feel will be basically identical. Well, tell you what, I'm going to transition now to my great sort of friend and colleague, Tyler, who's going to take over on the sort of hands-on part. But I'll keep an eye on the chat. I'll be here throughout the whole call So if you have any sort of additional questions, feel free to just drop it in the chat. And again, we're all here to support you through your analysis today. Thanks, everybody.
All right, guys, I'll take over for the next part. My name's Tyler Collins. I'm a member of Mike's lab, and I kind of will be going over two parts. The first is going to be kind of a high-level overlook of the metagenomic data we're going to be working with today, the project it originates from, and then the subsample data from a specific site that we'll be using. And it'll be a nice collection of metagenomic soil data generated from PacBio hi-fi sequencing. And then after probably about 10 to 15 minutes of looking at that, we'll I have screenshots of the entire process, but we'll do the live demo and walk through Galaxy, show off how it works, and run through some of these analyses. Some of it will be a bit pre-run. For example, some of the assemblies just take 40 minutes, 45 minutes to run, even on these smaller data sets. So it wouldn't be really feasible to try and do it live in this exact setting. So I have it pre-run, but I'll have all the data and histories freely available, and we'll show you how it all works and how you can analyze it and run through everything on Galaxy. All right, so to begin with, we're going to be working with soil data today, and you might ask, why look at soil metagenomic data as compared to the human gut or ocean water or anything else? And I think one of the main answers is soil is one of the most biodiverse habitats on Earth. There was a recent paper from 2023 that was making an argument that huge amounts of the diversity in life is actually present in soil more so than any other environment. In addition, a lot of the microbes in soil are unculturable in a lab, and so it's notoriously understudied. I forget the top number off the top of my head, but it's over 90%, I believe, are unculturable. And so there's so much unknown, and it's a super interesting place to try and look at and just make some new insights and discoveries. More specifically, from like a bioinformatics side of what we're trying to figure out here, we have a couple main questions we can really be asking. Simplest questions are, what's even present in the soil? What kind of bacteria, archaea, fungi, what's just present? And then not only what's present, but how is that genomic composition changing? Does it change over time? What are the metabolic pathways that are being expressed? What's happening? In addition, this also gives us an opportunity to answer some more interesting bioinformatic questions. Metagenomic soil data presents some unique challenges when trying to assemble and research. Very often you have tons of species and you get low amounts of coverage on many species, so it can be hard to assemble. So in the context of this broader project, we've been doing comparisons between short and long read sequencing technologies and several other methods and trying to find answers on better methodologies, almost like an optimal approach to solve this. Additionally, we're trying to answer some like broader, more useful questions for people. For one, how is the soil composition, the bacterial composition, our kill competition affecting like agriculture, the environment, and more specifically to humans? We've been looking for antibiotic resistance genes and other things that are present in soil. If you're looking at soil that's present in Baltimore, where Hopkins is, are you finding dangerous levels of antibiotic resistance genes being expressed that may have negative health impacts on humans in general? In addition to those kind of bioinformatic questions, we also can kind of consider some of these environmental questions. So some of the partners we work with have been saying the samples from Superfund sites. The top kind of images with the heavy machinery come from a site in Atlanta, Georgia, and that was sent to us from our partners at Spelman College. That's from a Superfund site where they're finding mass amounts of high lead contamination and led to an EPA major cleanup. And so when we're looking at these soil data, we can look at and say, okay, if we had these heavily contaminated areas, is that heavily impacting the microbial life that's there? And then kind of what's the effect of that compared to healthier land around it? Additionally, we can kind of try and answer some more generic questions of, are there significant differences between managed land versus wild ecosystems? Are we looking at big differences between urban and pristine sites? How much human impact is causing changes? How is that affecting differences in bacterial representation? And so in order to address some of these questions, this big data collective called the Genomic Data Science Community Network was started, and more specifically for our project, a sub-branch under that called BioDigs. This is kind of a large joint project that works with Hopkins and all these other partner colleges, that works with sending out students, undergrads, graduate students to go collect soil samples from localized areas. You can see on the map to the right here, we're getting samples from all over the United States. Those get sent back to Hopkins. We do a series of testing for, or I should say, get sent back to Hopkins, and we send the University of Delaware and a couple other partner institutions as well to do soil sequencing, test for heavy metals, pH, get a bunch of metadata, and then make that data accessible for some of these metagenomic research projects that we're working on. For biobigs more specifically, here's just an example of some of the sites that we currently have where we have sequencing data and heavy metal data. So it's just showing off a little bit of the range of the country. We have several partner colleges sending that information to us, and we've been pulling in lots of heavy metal data, as well as other metal and soil metadata. However, some of the most interesting findings, just so everyone's aware, there's been quite a bit of above EPA safe lead and arsenic limits that have been found within city limits in some of these areas. And even just without any of the sequencing, there's already been some quite interesting and slightly concerning health impacts. But in addition to that kind of metal metadata and soil testing, this project is also doing a lot of some of that sequencing. So the kits are sent out, people harvest the soil. It's protected with something called Zymo DNA RNA shield. This just ensures and helps prevent the breakdown of the DNA and RNA during transport. Once it arrives, it usually goes through, we've done it through 3 sequencing methods right now. It's been a mix of Illumina, PacBio Hi-Fi on the Revio machines, as well as some of it's been done through Nanopore for a mix of long read and short read sequencing. And then that data, that sequencing data, is what we'll be looking at today. And so specifically, I have on the slide, I'll say, the Biodigs website, if people want to go look and see the full sample mapping, learn more of the data. But for our test kit today, and what we'll be doing for our live demo is using a sample of sequencing data from Gwynn's Glade. This is a kind of like semi-urban wild area that's kind of next to a freeway. It's like, I guess you call it like an urban greenway, might be the best descriptor for it. That's in the southwestern side of Baltimore. This was one of the pilot sites that was sampled for the Biodigs project. So it was one of the earlier ones. But we have some soil data that was sequenced for both long read and short read. And it will kind of represent kind of like a not disastrous, super contaminated site, but kind of a nice baseline for us for our analysis later. So here's some of that metadata about our specific site we're looking at. The top graph is going to be looking more at the heavy metals. The bottom one is going to be looking at more of like random nutrients. The top one's annotated with the Canadian agricultural guidelines for safe levels of metal and soil for agricultural products. The EPA and the US's are a little less strict often, but sometimes a little harder to actually find exactly what it is. And I'll also point out arsenic, if it's below 30 milligrams per kilogram, is not detectable with the current tests. But when we look at our site here, we can see that all of our heavy metals are about in the acceptable limits. And as far as most of the sites we've been looking at the US, pretty good, especially compared to the heavily contaminated areas. And if we look down at our random nutrients and metals, we see kind of a nice spread with lots of them being present, but no like super high domination of any of the nutrients, although calcium is a little high. In terms of our sequencing data, this site received 2 types of sequencing. So it used short read Illumina sequencing that was sequenced in Baltimore. And then it also used a PacBio Hi-Fi Revio sequencing. The Hi-Fi Revio is what we're going to be using today. This is just long read sequencing. You can get about, I think the average for this data set is about 10,000 base pairs for the average read that came out. So significantly lower than the was 150. And that will matter a lot for helping us be able to get better assemblies and try and do some taxonomic identification. Slight spoiler, I guess, for now, when we try and do the same analysis on that Illumina data we had originally pulled from for like identifying which species, about 90% of the reads weren't able to be mapped back to a species when we use that PacBio Hi-Fi data. Today, about 0.02 or 0.08% didn't map back. There's a couple of caveats there, but we can discuss that as we actually get to that in our analysis later. Finally, we're also going to be, or not finally, but we're also going to be subsampling those reads. So this is just some of the protocol. We're basically taking a 10% sample of our read data from that hi-fi. We're just doing this to reduce the data load runtime. The bigger your sample, the longer things take to run. In theory, this should save the same coverage and same species. We've been testing to make sure that it still provides good enough results, but this step is mostly just to save us a lot of time. But if you're interested in the actual commands, we're using the CTK sampling. We had a set seed. The seed was 42 to extract these reads. And so all this is doing is shrinking our data footprint, especially if we want to do some like early analysis before running through everything. All right, and so in terms of our plan for today, we're gonna load into Galaxy and we're gonna kind of load our data in, run some kind of quality assessment metrics on it just to ensure that the data is what we think it is and that's working correctly. We're then going to create an assembly. So we're going to take all those raw reads, run it through a tool, generate contigs. So we're going to try and stitch them together as well as we can, see what we can get out of that. While we do that, we'll also do some kind of assessment on our contigs. We'll show off some of the visualization methods you can look at it, of advantage plots, how to view an IGV, some of the coverage, and discuss some of the biological implications of what we're seeing there. And then finally, we'll kind of move on to some taxonomic profiling, trying to see, like, can we assign these reads to certain bacteria, certain species, and what we want there. And I think, yeah, sorry, I just saw the chat, but I will come back to that in a second. And I just also want to point this out just because I think it's always useful. When you're doing this kind of work, there's some important challenges to keep in mind that can make the results harder to interpret or cause problems. What we do today, I've run, so I know it will work. It will work well. But if you're actually doing this on like your raw data or anything, especially if you're doing soil data, you need to consider that soil data is very diverse. Very often when you do the sequencing, you'll get low coverage of tons of species. Some will be related, they'll be closely related, you'll have small mutations, and this can make assembling and binning have increased error rates. Also, when we're doing this like taxonomic assignment, doing that is very dependent on the quality of the database for the tool we're using today, on how much like genetic knowledge they have, how many genomes they actually have stored in their database that we can compare against. And changing that can significantly change your results. And so these are just a couple of the things to consider. There's definitely more complexities, but this is just to point out that there'll be a lot more problems that'll arise, especially if you're doing this with your live data. And so with that, I guess I'll take a moment before we move into the actual live demo and go to the platform and walk through how to run all of these to see if there's any other questions in the chat and see if I had missed anything.
I don't think there was any questions in the chat. I know that Dr. Graves did post a scientific journal article about antimicrobials in the chat. I think Dr. Schatz answered everything else. Before you get started, I would like to say that, I have put a post survey in the chat for this workshop. If you please could fill that out, it is great for our presenters today and for GRADS-4C, just for feedback.
And we'll continue. So this is kind of the overview of what we're going to be doing in Galaxy. I'm going to be hopping in between these slides. I have screenshots of everything that we'll be doing that we'll kind of walk through, organized by the main sections. I'll come back to kind of these overview slides whenever we hit a checkpoint. But other than that, I'm going to be doing the crazy thing of trying to do this live and make sure it works right and just hope nothing goes wrong as we run through this. So I guess I'll also take the moment now to see, I know Mike mentioned earlier about creating an account. If you want to follow along, just make sure you're logged into your Galaxy account. I will start sharing the links of how to import histories or move data in if you would like, to run these analyses along with us if you would like. And that will probably occur here after I show a little bit of the beginning parts of Galaxy. So with that, and seeing no other questions, I will start us off. All right, so welcome to Galaxy. This is kind of your default landing page of what you're going to look like when you enter the platform. Whenever you log in, this is what you'll see, and you're going to see it kind of consists of a couple key parts that I'll go over. So on the left, like far left-hand side here is going to be something called the activity bar. It's reminiscent to VS Code, if you ever used it, or any like coding editors. The point of it is your like large activities are organized in it, like main things you'd want to reach. So for example, it gives you the ability to upload your data would be something you could do here, to search through all the different tools, scientific tools that are available on the platform, to search through all the workflows or create your own workflow that's available, to see which workflows are running currently or which ones you've previously run and see the results, to try and visualize your data in different ways, and then a couple others that we can kind of get into later. In the middle section here, where you'll see kind of the scrolling carousel with information, this is going to kind of be your workspace. So whenever you open a tool form, whenever you're looking at visualization, it's going to populate here in the middle, and you'll always have access to this activity bar. And then here on the right is going to be your history section. Right now it's going to be empty. You can see we have a brand new history. And in here is going to kind of track along what we do, and we'll store all the information and kind of populate down here, showing step by step what we've done so that we can reproduce it if we want. The last thing I'll point out, and something that I think is always very useful, is you'll always be logged on the right like you think you will, but you'll have a whole set of user preferences. When you click on this, it will give you the ability to change your password, delete your account if you so desire, change themes, and then access some very useful other settings. If you want to try and connect third-party like cloud buckets to pull information in, see your API key, address dataset permissions. These are kind of a lot of the more advanced things. And as well, in Galaxy, you'll have storage up here. By default, we provide all users for free 250 gigabytes of storage they can use forever, and then 1 terabyte of scratch storage that will free itself up after about a month of it being used. That will exist in the scratch storage will remove itself, but 250 gigs will exist in perpetuity. But if you also ever need more for a project or something specific, you can reach out to the help team. They can also often give you more storage. This is just to help maintain our resources, although I tend to have a lot more storage, I guess, than most other people. All right, so with that said, probably the most important thing you want to do in Galaxy before you do any form of analysis is figure out how to upload your data. And Galaxy kind of has several main ways to upload your data. So I'll just hop back to our slides for a moment just to illustrate it before going through it live. So there's kind of 4 main ways to get it in. The first is something as easy as a local upload. You can literally drag and drop a file off your desktop, anything on your local computer, you can immediately pull into Galaxy. It can fetch data. So if the data is stored on some website somewhere, it could be Zenodo, it could be something else, you can put in the web request, Galaxy will make a fetch request against that, pull the data in and install it. If you have your data stored in Amazon Web Service Cloud Bucket, or if it's available like Microsoft Azure, you can enter your cloud credentials, and you can pull data directly down from that into Galaxy. And then lastly, if you are using a university like supercomputer or an HPC or something, there's a Python or a Pythonic package of Galaxy called Galaxy Upload, where you enter essentially one line and like your API key, and you can transfer data quickly off your university supercomputer or HPC into a Galaxy instance, into a direct history. So those are kind of the main methods. So I'm going to kind of show you how to bring it in, and then I'll start sharing histories after we have something available within Galaxy. So easiest way to bring it in first is if you just click this little upload icon up here on the top left. Will open this kind of little modal window here. You'll have a couple options. The first one we're going to look at here is just choose local files. In here, you can open it and you can just see all the local files that you have available. It's as simple as this will be, I guess, a banner image. This will just be a PNG. It will load here into the window, and then I've selected it. All that I have to do is click start. It will start uploading it, and then I'll close this modal for a moment. And you'll see in the history here, it will turn gray. When it starts processing it, it's going to turn this nice yellowy orange color. And then once it finishes and the data is actually available to me, it's going to turn green. So we can look at that in one more second. If we come back to our uploads, in addition to doing it locally, very often a better way to get your data in is to do that web-based thing. Very often genomes and things are stored at web endpoints where you can request and pull them in. That's as simple as clicking this paste and fetch data. It's going to give you this little box you can enter. You can enter a single URL per line, and we'll go out, try and pull those and bring them back in. So I believe I will find our link, but it might be down here. Yeah. Show you what that looks like. So this is a Zenodo link to the live data set we'll be using. I can just paste that in here. This one's a couple of gigabytes compressed, even downsampled, so it'll take a little while to pull in. But something as simple as that, you can hit start. It'll pull it in, and you'll see it start to fill in here in the history in a moment. Trying to go pull that in. Other things to consider. By default, it will just name it whatever it was previously called, but you can assign a name immediately when you pull it in. And it will by default also try and auto detect what format your data is. I'm sure many of you have known through bioinformatics, there are tons of different data formats, hundreds of them. Galaxy supports the vast majority of those and has coding to try and find whichever one it is and then pull it in. I see a question right now. You can upload any of those. Galaxy supports, I believe, almost the vast majority of data types. You can see through here, there's like hundreds in here. We'll support PNGs. These are all random bioinformatic data types, especially a lot of mothers, FASTQs, different gzipped files, and more can always be added, especially if new data types are created. We'll support zips, TXTs, tabular, PNGs. Any of that can be pulled in. HTMLs, any of that. So those are kind of the two easy main ways to usually pull in. I was also going to take a moment to briefly show how you would set up a cloud bucket, just because very often if you're working on a bigger project, people tend to like to store that, especially if it's between multiple universities, somewhere in the cloud. That's as simple as if you come to your settings, you can come down here to manage your remote file sources. If you click that, this will just show you a list. Oh, yes. So I saw the link to the demo data. I can share, the link is on slide 55, but I can also link it in the chat if people want to pull it in directly.
Yeah, I think that would help if you just dropped it in the chat.
Yep, I just linked it in the chat, so everyone should have access to it there. That'll take a little while to download. I also have the histories saved, so we can import those, so everyone can follow along easily too. But I'll pull those up in a sec. But yeah, so in terms of your remote configurations, you'll have something like this. To add a new one is easy as clicking create. You can select any, right now we just have AWS, Azure. I believe Google Cloud is coming shortly, but I think that might be with the next release, but I would need to confirm. But you come here, it'll just ask for your name, access keys, information about it, and then it'll allow you to create it. Once it's created, it's as easy as accessing. It's always coming to your upload. You can click this choose remote files. This will give you a list of all the remote files that are publicly available, as well as your AWS ones. You can just click in here and you can navigate it like a regular file directory. Oh, as I click on a bad one. I don't know if I have access to most of these, but you can click in here and you can see all the files. This will be organized like a file directory. You can click these, select them, they'll bring it in here, and you can then upload it into your history as well. So those are kind of the three main ways of bringing things in. I'll look briefly in the slides. This is that fourth way. I have a link to our documentation on how to do it, but if you want to bring it up through the command line, the Galaxy upload utility, using a command like this is the best way. Basically, you just run the utility, say which Galaxy instance you want to go to, pass in your API key, which will be accessible in your settings menu, and then the name of the history you want to send it to, and it will upload it all using the TUS protocol from whatever remote server onto the Galaxy platform and give you access to it. In addition, so once you have a little bit of that data uploaded, you will be able to see it kind of turn green here in your history. You can see the small PNG loaded really fast, the bigger data sets still coming in. But you can click on these items and it'll kind of drop down and show you what you can do in it. Here you'll have a couple main things. You'll have sort of the size of the file, what format it's in, its name, and you can click this little eye to like get a quick look inside the file to see what's happening. So in this case, you can see there's our PNG. We can see it here in the middle. This is for a graphic for the upcoming GBCC conference for the group hackathon that people will be doing. In addition, you'll also be able to click on this like edit dataset attributes. And if you so desire, when this fully loads in, this will allow you to like change the name. So we can change our name of our dataset here to whatever we want, add a custom annotation if we want, and we'll be able to, we won't cover it today. We can adjust permissions of who can access it, what can be done with it, and then you can also change the data type. So if this wasn't actually a PNG and Galaxy guessed wrong, or I mislabeled it, or something happened, I could tell Galaxy this is actually a different data type. So with that, I will also share here on the slides. So there will be kind of two ways to do things going forward. I have a series of separate histories kind of highlighting these different steps. That bottom Zenodo link is a link to the raw downsampled sequencing data we're going to be using. That above link is a Galaxy history that I've published and shared that anyone can use. So if we just, thank you, Natalie, if you want to paste that in the chat. So if you want to follow along live in your own Galaxy instance, you can just copy and paste that, and then it will basically say, do you want to import this history into your Galaxy account? You can say yes, and it will load the history here into your little intro. And that history will be this guy, which is just going to consist of one file that we uploaded. This will be our subsample data. So now we've gone and uploaded our data, no matter how we did it. First step is let's just kind of take a look, do a little sanity testing, make sure we have the right thing. We can come in and see that this is about 3.6 gigabytes of compressed data, which makes sense. That should be about the size of the file and was the same size of the file in that remote storage it was located. Additionally, it will kind of show the first few lines of the file. We know this is a FASTQ, so we kind of expect a sequence identifier, the sequence, a spacer, and then some kind of like quality control. And we can see a sequence identifier here in the sequence. To get a better look, we can just click that eye, and it will kind of load it here in the middle. On particularly large files, it will only load a small section just because loading millions and millions of lines will inevitably cause performance issues. You can force that if you want. I would recommend not doing that. This is mostly just that quick look, and we can see, hey, look, here's our identifier sequence with only the 4 nucleotides we'd expect to see, some kind of spacer line, and then our quality scores, and then the next one repeating again. So this would look like the correct kind of file. And with that, that's kind of, I guess, the first steps in how you upload things in or pull in data, start to look at it, and like a little bit of how the history works. So following that, even though I just had you guys import that history, I guess have you import another one. Now that we have our data in, one of the first things we want to do is some kind of like quality control, take a look at it, do some sort of light analysis. So I'll paste this history in the chat. It'll be the same thing. You can click on it, import it, pull it in. But we'll kind of go through some quality control and then some interpretation to make sure that subsample data we pulled in makes sense, that we have good quality, and that it's working well. So here's that subsample data we had again. Will be the same file with the same kind of view. And the very first thing we're going to want to do is a very common quality control tool you'll see on FASTQ files is something called FastQC. There's several others available on Galaxy, but this one kind of makes a nice little web page that I figured would be nice to look at. So I'll just kind of show you how to access it, what it looks like, and we'll kind of go through and look at a bit of the science together. So to find any tool in Galaxy, you can just click the search and run tools, and it will open this little toolbar right here. You'll see it's kind of organized into different categories if you want to use, but there'll also be a search bar here at the top. Usually when I use it, I only ever really use the search bar to find what I'm looking for. In our case, we can just search for something as simple as FastQC, and we can see it'll be right here at the top. And if we click that, it will open what's known as one of those tool forms in Galaxy. So this represents the FastQC tool. This is kind of a graphical interpretation of that command line that Mike was showing in his earlier slides. I do have a workflow for it, which we will be showing it near the end. I'll show you how to make one and then how to actually pull it all in and run the full workflow yourself if you'd like. So how this form works is you'll have kind of like a run tool button here at the top, as well as a version history. Galaxy will maintain every single version of this tool that's ever existed on Galaxy for reproducibility purposes. By default, it will show the latest one, though, the most up-to-date. You can see here we have raw reads from data, so we need to pass in some kind of raw reads. Right now, it will show that all available ones from our history that like have the right file format type that could be entered here. For our case, this 10 is the small subsample one we uploaded. This other one up here is an even smaller subset I did for an upcoming tool to show, where it's down to like less than 1% of the total reads. So we can just click here. This will reference this subsample down here in our history. We'll also have some optional parameters. We could pass in some kind of contaminant list if we want, some kind of adapter list, look at some kind of limiting. We have a bunch of other options here we can come down and pick from if we need. By default right now, we're just going to do the regular default options. We're not going to mess with anything. We'll also have our run tool button down here with the ability to be emailed if we want an email notification when the job completes. FastQC will run pretty quickly. Even on this data set, it might take a couple minutes. But the longest jobs I've seen running on Galaxy are some of the, like, if you're doing like telomere research and things, jobs can take days to complete. So very often, something's gonna take a long time. It can be useful to get an email if those jobs complete, or if you know something's gonna take hours and hours and hours. And kind of the other thing I really want to point out in most of these tool forms is every single Galaxy tool form will have a help section. Not all of them are perfectly fleshed out. It depends who wrapped the tool and added it into Galaxy, but most are pretty thoroughly. And very often what this will do is it'll tell you exactly what the tool's doing, what you need in it, what its goal is, what you should expect as an output coming out. It will also show which tutorials use this tool if you want an example of someone else using it with some other kind of data. There'll be a link to the Galaxy help forum where people from Galaxy answer questions all the time. So there'll be other questions people have been asking about this tool or having problems with it, or if they need help as a good resource. You also have a link to go ask a question immediately if you would like it. And then finally, you'll have the actual citations to it, and usually a link to the original GitHub repository or other spot where the tool is, if you want to go find the tool and run it locally or see the actual source code behind the tool. So for our case, if you have the subsample data in and you're doing it that method, you can just hit run tool here and it will start running. I guess I will also run it. There's no reason not to. It'll just populate inside this history. You'll see it turn gray, and then it will turn yellow as it starts running. And then once it completes, it will turn green. Just for the sake of our time to run through more stuff and have time for questions, I've pre-made all this. So we have a completed run here, which should be the same as these. And that will be these two outputs here. We'll have like a web page output, which will be a pretty graphic, and then our raw data if we need that for some reason. So to start with, you can see our web page. We still have sort of the same options here. You can see we have an HTML file of a certain size, but we just need to hit the I here. And we can kind of see it in the middle. And here we have our generated FastQC report on that subsample data. So at the top, you'll have some regular information. You can see our total sequences we had, the total bases we had. But we'll kind of walk through here and explain a bit the biology. So I'm guessing most people have seen FastQC reports and done some kind of quality control. But if you haven't, basically things in the green and green check marks are usually a really good sign. Yellow are slight pauses, and red is something you should probably investigate. In our first graph here, we're looking at kind of a per base sequence quality. So the bottom x-axis will be the actual sequence position of the different nucleotides. So it'll kind of scale in a weird way, but you can sort of see kind of like averaged out in these different bins what the average quality score is. And that quality score is basically an estimation of how likely there is to be an error in that specific base call. At this high level in green, we're expecting incredibly few errors, and it's very good. And you'll kind of see as our reads get longer in their base pairs, as like the base pairs get longer, the quality starts to diminish a little bit, which is expected. But for the entire time, you kind of get a larger variance, what we're seeing in the green. So for the moment, it's pretty good. If we were more concerned about this, we could do some additional trimming, try to remove bad reads, do a couple other things. But right now, we feel pretty good about that. We kind of get a second graphic view of what that looks like. You can see the mean quality score over all of our sequence of what the average quality score is. And the vast majority of them here are living at a peak of about 39 and looking very good. One of our first things you might see that got flagged here is actually our per base sequence content. It's flagging that because there's a divergence. If you look here in the red and green is going to be your T's and A's, blue and yellow C's and G's. And in our sequence data, we pulled down about 60% of all the nucleotides are CG, with about 40% being AT, so it's a little skewed. And if we kind of look at it more at a specific level, you can see kind of a very high peak of GC on our theoretical distribution. So some of our reads are having super high GCs. Another interesting thing to note that we won't go super into is you sort of see a little sub bimodal peak here. We won't fully dive into this, and I personally haven't gone deep enough into this subsequence data to fully identify why there's such a GC bias. It could be something as simple as the extraction method was slightly favoring pulling that out as compared to other methods. It could just be true biological variance within the soil, or it could also be something wrong with the extraction protocol or some of the analysis and cleanup that we've done so far. But outside that, the rest of this we'll kind of briefly glance over. We don't have any unknown bases really. It's kind of our sequence length distribution. So the majority of our sequences are probably somewhere around 8,000 to 9,000 reads with a couple super long ones down here at the end and a few short ones. And then we don't have high amounts of duplication. And then for our cases, this is just a default adapter check to make sure there's not leftover bias from the actual sequencing run. And if we had Illumina adapters in a PacBio run, I'd be concerned, but there's none, so we're looking pretty good. That's kind of the brief over FastQC. There's a bunch of tools that work like this in Galaxy, where you can kind of get a visualization here in the middle, check your data, because realistically, trying to look at tables or information like this, you're gonna get nothing out of it. So very often it's better to visualize. I also wanted to take a moment here to show off another kind of tool that shows up in Galaxy and a slightly more fun one to look at that same information. So this 13, this subsampled smaller subset tag here, I'll show off. You can add tags into data sets just for splitting things up and understanding better how it works. So you can just do like some kind of pound or whatever, and you can say like, this can be training or call it learning. And it will add this tag. So in your history, you can have tags to better organize, especially if you get very long histories. This can be a good way to help differentiate things. And you'll see people add these by default in a lot of workflows so that when you run the workflow and it fills your history, it will auto add tags to make it easier to navigate. But the point of this is we did a heavy subsample on this data so that we could run a program called FastQE. FastQE take a little bit longer to load here as compared to FastQC, although you'll soon find out why. It's another method of looking at quality control. Another method of looking at quality control in our data. And rather than those beautiful graphics, this is a program that somebody wrote that analyzes each individual base in your reads based on emoji. So depending on how happy the emoji looks, it scores it based on how well or how good your read was. So you can see in our case, in this blinding yellow array of never-ending emojis, we have lots of very happy ones with one's quality score here of a broken heart that's really bad. And you can see as we look across our reads, they'll tend to get a little worse as it goes on here at the end. But overall, for the most part, very high levels of reads. In terms of the metric of what you want to see what it looks like, they kind of have their scoring methodology here. So yeah, that's kind of our initial looks at quality control. Basically, when we look at this, we see that our data Pretty high quality, not high amounts of error, and like pretty good to look at originally. So with that, we can kind of move on to our assembly and visualization steps. And this is kind of where more of the real analysis will actually start on this. And that's the link to the history if people want to import the assembly and visualization history in chat. So I'll move us over to our new assembly and visualization history. You should see here on the bottom our subsample data. We still have our FastQC data, and this is where we're going to introduce our assembler we'll be using for today, which is an assembler called Fly. There's several metagenomic assemblers that get often used, MetaMDGB, Metaspades, Fly in metagenomic assembly mode. For this case, we'll use Fly. MetaMGB is currently not on Galaxy, but is being wrapped and being added into it. It was just pending a couple problems that were showing up, but we'll kind of walk through here how to do it. And then we'll kind of go through our data, and then we're going to show off some visualizations to try and really look at how our assemblies came out from when we ran this. So much like the FastQC tool and most other tools, there will usually be some kind of like data input you need. In this case, it will once again require some kind of FastQ or FastQGZ or any of these you can see in the acceptable formats here. So to run it, for what we have done is we've just chosen our subsample data here, which would be that 10% subsample of our original data. We can come down and look at our mode. This will consist of all the different sequencing technologies for which we want to pick from. In our case, we're using PacBio HiFi data. When you select this, you'll see it will change some of the options here. It'll update on the fly to match whatever mode you're in. In our case, we're just going to rely on the defaults. For the error rate, we're going to assume error rate of 0.01. We're still just going to do one polishing iteration. We'll keep the minimum overlap still as a default. We're not going to do anything with haplotypes for the purposes of this, nor will we change enable scaffolding. But what we will do is we will specify that this is a metagenomic assembly. This is one of the options within Fly that will basically change how it operates pretty differently, because it will now assume there's very non-uniform coverage amongst our data. Certain species are going to be heavily overrepresented, some are going to be incredibly rare, and it will vastly change how this program will run. Additionally, we will disable the reduced coverage here, and then we'll generate a log file. And the purpose of the log file is just to let us see exactly what is being outputted by the tool standard output that's going through it live. And then from here, we just need to hit run tool. And once we run, I've already done this once again, you'll see it should take about, I think, 30 to 45 minutes, depending on how resources are being allocated, how people are trying to run it. If you guys try and run it right now, you'll see we get about 5 outputs. So in our history, we'll have this fly on data 9 consensus, an assembly graph, a fragment assembly, some assembly info, and then the log I was mentioning earlier that we added. So if we look at the consensus file here, the consensus file is our config file. We have about 1,917 contigs in it. Here's an example of the first one, kind of scrolling down. You can see it being stick together from reads. But ultimately, much like most things, this isn't particularly useful without more information. The other thing that Fly will give us will be some kind of assembly graph thing here, which, sorry, let me show the graphical assembly, I guess, which will give us all the edges in the large complex graphic structures to make its contigs. Once again, hard to visualize, not super useful here, but we will be able to use it here in a moment. What is useful for us, though, is Fly will automatically generate some assembly info. By default, this will order our contigs by the one that's the longest. So we can look in column 1 here and see the sequence name. These will just be our contig names, the length and base pairs. So our longest contig that we generated from our subsample is about just under 600,000 base pairs, which very likely is a complete genome of one of these microorganisms. It will also then tell us kind of like the average coverage of what we expect in that contig for how well it's doing, and then some other information. So this will kind of give us our first indication of what we're looking at here. However, I often find it's very useful to visualize things. So one of the ways to visualize how your contigs look that used to be more common for the Illumina, but is still done a little bit with some of this hi-fi data, is to do something called a bandage plot. So we can go find this tool bandage image. And if we look in the bandage image, we can pass in, the graph. We can pass in an assembly graph from Fly and basically specify how big do we want this image, if we want any fonts, the file type we want to come out. And what it will do is it will basically run through, build that graph, and then try and construct a visual representation of what that graph actually looks like so we can look and see. Very often you used to get graphs that looked kind of really messy and convoluted like some of these. That shows up often with short read data, especially older short read data. You can get some really gnarly graphs coming out that can be useful to visualize your contigs and their quality. However, since we're using longer read data and some of the hi-fi data, after we run it and we generate here, we can take a look and we can see our bandage graph is a lot more boring. What we're seeing here is this would be our longest contig here, and you're seeing kind of just long uniform contigs that kind of get smaller and be of different sizes without much variation. So there's not much disagreement from the contig about what's actually splitting off. The two examples I would show where you can see some of that is if you look at these two kind of antibody looking ones down here, we see them forking off. Those will be kind of your unique splits. This is mostly just kind of that immediate look. You can sort of give a visualization of your graphs, what your contigs look like. And it's kind of a nice sanity check that I would often like to do after doing some form of assembly. In addition to very often other things you do after that assembly is you run a lot more quality control metrics and some other things. You might want to run Quast. You might want to maybe run some, like, if you think you have a complete genome, some kind of like Busco analysis to try and estimate how complete of a genome you have, things like that. But those will be a little out of the scope of at least the demo of what we'll be running through today, just for time purposes. So with this assembly graph, we might think, well, let's also try and visualize our coverage. Let's try and take a look, and if we want to see our contigs, how many reads actually map to those contigs? What do those actually look like? Does it make sense? Is there something wrong? More sanity checks. So to do that, we're going to use the IGV, which is a nice genome viewer. I'm assuming most people are familiar with, or if not, is a very common way to try and investigate visually how a genome is structured. So the first thing we need to do is generate a BAM file, because when I moved it down, I did not move the BAM file with us. So we can do that. I guess I'll also show here. Whenever you run something in your history, just as an aside, you can always click this little like round Ouroboros looking arrow. That will be a rerun, and it will basically open the tool form populated with every single thing that was used to generate this data, how it was exactly before, so you can see exactly what you used. So very often if something goes wrong, you need to rerun something, or you want to see exact options you chose, this is a nice way to be able to do that. In this case, we're going to be using a tool called Minimap2. We're basically going to be passing in some kind of consensus sequence. So this will be our contigs that we generated. And we're going to pass in our original subsampled FASTQ data. And then we're going to basically tell it, hey, these are hi-fi reads. Please consider that when you're running this. We won't, in our case, be adjusting any other options, but you'll click down here and you'll have full access to tons of other options if you so desire to customize this more and more, if you want to change clipping or cigar outputs or whatever else. For our cases, we're just going to keep it a little on the simple side. But when we run this, we'll generate this BAM file. And from that, we'll actually be able to do something cool with a really nice visualization. So to visualize what we're seeing on all of your history options, you'll have a couple little icons down here that you can pick from. One will be to download it from Galaxy locally. But for our cases, one will be that rerun I said, and then this one will be visualize. So if you click this, it's going to open up in the center of the panel two things. One is we can display it with these different links, and then one will be built-in Galaxy visualizations. I'll say right now, in May on the next Galaxy release, there's a big overhaul of the internal visualizations that drastically improve them and allow way more complex ones. Right now, most of them work fine, okay, but I think in about, I don't know, 4 to 5 weeks, you should see a lot of brand new ones that are significantly more complex, especially on the spatial omics side. But for our case, we're going to be working with this and showing how it works with IGV. So we have the option up here to display with IGV locally. I have IGV installed. There should be a link in the slides if you want to pull it down locally and install it. But in order to show our Galaxy data in it, we just need to click this single link to display locally. It's going to launch this little window. That won't matter. But I guess I'll need to share my Zoom difference too. That will take our BAM data in. The other thing we'll want to display is we will want our contigs displayed locally. So we can go to our consensus file, also come down to this visualization, and then we can also click display with IGV locally to have it display locally within there. And I will change my screen. To display IGV locally. All right. All right, so this is the IGV displaying locally. Up here in the top, you'll see where it says like Galaxy and then all the random like jumps and letters. This is going to be our contigs that we generated. And you'll see we have access to all of them. They're ordered in like the programmatic way, not necessarily the regular human counting way, but you can search through all of them and every single one will be present in here. In addition, we'll have our BAM data kind of down here on the side on the track. So if we want to take some early close looks here at contig number 1 of what we looked like, we can zoom in kind of here at the start, the original base pairs. We won't go super in depth on how to stylize this and look at things, but here we're looking at kind of like an 832 base pair region of that contig, starting from about 5,800 base pairs in it to about 6,700. And you can see here will be our actual sequence that was declared down here at the bottom. And then this will be kind of our coverage graph up here on this top one. And this lighter gray here will actually be the individual reads that mapped. Where it's all light gray, that's where everything matches and is uniform and it's not particularly interesting. We can click one here and get a little window that pops up and tells us information about it. And then kind of wherever you see the different colors or little gaps or anything is kind of where some differences occur. So when we look in this region, it seems like we have 4 reads mapping to this part of the contig. That's maybe a little low, but this is also a 10% subsample of our original data. And it's pretty uniform, only some small differences. So it looks like a pretty successful map. But as we scroll along, we can kind of see we have our little coverage graph here at the top, mostly the same, and all of a sudden we hit this big section. I'll even zoom out a little. We hit this really big section where the coverage spikes up and we get tons of reads, all mapping to the same thing. And if we look at this, you can see there are tons of random SNP mutations. There's definitely some deletions, some other contigs mapping back, some insertions. There's a lot more going on here, which is one of the areas you'd want to consider and really look at more in depth. Something weird is probably happening here. Maybe it's, if you look at kind of some of these more individually, like especially these top 4, they all kind of have the same mutation pattern. Maybe these are all coming from the same species, and some of these are from closely related species with a couple changes. Oftentimes, the assembler is going to try and do a better job of sticking things together. It will favor that over other things. This would be something you might want to look at, especially with this contig. The other contig I think we'd briefly look at here is 1406, which was our longest, most complete contig that we got out of our results. And you can see here, this is our one that's a little right around 600,000 base pairs long. And you can see we also, even in here, if we zoom in, have some weird, some of those similar coverage maps. And like, especially in areas like this, where something crazy is happening, would be a particular areas to look at and try and investigate more for quality and see if you need to go back and change some of your assembler settings, see if you need some more fine tuning, something else like that. But that's kind of the display of, go back to the Galaxy now, of how it kind of works with IGV. Sort of showing how that integrates and how you're able to run with it. And with that, that kind of takes us to our next step, which is going to be the taxonomic assignment. So I will link in that new history into the chat if people want to import it. But kind of the goal of this step is we kind of skipped ahead a little, and we're sort of skipping some of the more quality control and trying to build like metagenome assembled genomes and doing some annotation and things. And instead, we're going to move and look at, from our reads and from our contigs, can we try and like assign those to a specific known species that exists in the world? Can we try and get some idea of what's present, the makeup of bacteria, what eukaryotic DNA is there, what's happening? So we'll hop on over to our taxonomic assignment. Ignore the error. We'll come to that in a moment in the top of this when you download it in. And we'll see we still have all that same history information pulling in, but we'll have something new. And the very first thing that we'll have new is going to be something called Kraken 2. So Kraken 2 is a tool that's designed to try and basically make that taxonomic labeling, trying to assign that sequencing reads. In our case, we can pass it in the contigs or the raw reads. For our use case, we just passed in the raw reads for the example today. And we'll basically be able to come down here and choose, do you need certain confidence thresholds? Do you need certain base qualities, minimum group hits to like kind of make it more sensitive? The two most important things for us to consider are, one, in order for us to get a nice visualization, we really want in this creator report to flag this aggregate count and clades to file choice. That will come in handy for showing off a really cool Corona plot here that we'll make. And the other most important thing with Kraken is the database selection. So Kraken will essentially break down your input reads into a series of small k-mers, which are like small partial reads that move across, and try and compare them against a very large database. How successful it finds matches is very dependent on how good of a database you're comparing against. Right now in Galaxy, we recently updated them to include many of the latest versions from their latest releases. But you can see here, you can scroll down, we have quite a few different databases available to pick from. The largest one we have right now is this GTDB version 220, as well as this Core NT that are some of the biggest ones. These are very large, and oftentimes if Galaxy is under high load, can sometimes get you in a little bit of a wait queue. The GTDB V220, I believe, takes up 500 gigabytes of RAM on its own to run before any of your data is put in. So very often you can start easily hit, 800 gigs, a terabyte of RAM, which is a significant amount of computing resource. But luckily, through our great admins, especially through Nate, who helped get all this set up, we're actually able to run it and be able to have people go through it. Just often, if too many people are trying to do those massive databases, it can get a bit of a queue to ensure there's enough high memory nodes available. For our purposes, though, we can select the Core NT for the run, and then we can come here and hit run, and it will take probably, once again, like half an hour on the size of this data to run through and give us an output. From that output, we'll kind of get two different things here that will be generated, a classification as well as a report. The classification, when we look at it, kind of gives us this nice useless table, but it'll kind of say a taxonomic ID and a little information. This is also about 1.3 million lines with tons of different entries and is not super useful to us. What's nicer to look at is that report we had generated instead through that extra flag we had. And this will essentially create a nicer, slightly nicer human readable version of the actual species with some taxonomic identifier information. But even with this, it's still not super useful for like interpreting our data or trying to understand what's there, really moving through it. So the next thing we can do is we can try and visualize this with Chrono. In order to do that, we need to modify this table into a slightly different format to be compatible with Chrono. So there's a tool in the Kraken tool suite in Galaxy called like convert Kraken file to Chrono text file. And basically, this lets you take in that tabular report file that we generated. And when we run it, it will do a pretty quick conversion where it will basically output it in a slightly better classified way that Chrono will be able to interpret. So when we ran it, that will be here in item 13. You can see that table we had before kind of becomes a bit clearer, where we have still a number count, and then you'll have a kingdom column, a phylum column, a class, order, family, and we'll kind of go down showing the counts of each as we move. So once we have our data in this more useful format moving through Galaxy in that way, we can actually finally visualize it somehow. So from here, we can go to Chrono Pie Chart, which is a very, very cool interactive visualization way that shows how it works. We just need to pass in the, we'll specify that's tabular, pass in our converted tabular file that we had generated at 13. We can leave everything else the same, and we'll get an output that looks like this. And it's at this point that we'll actually get a better feel for our data and our taxonomy. So when we run it, we convert that long, multiple millions of line file into this really cool interactive chart that essentially allows us to move between those kingdoms, phylums, clades, and have it move around and look for more interesting things. And additionally, we can take snapshots and things if we want to save things to have it be easily exported or moved and adjust a few things. But right now, upon load in, this is just the root level of the file. We can see the vast majority of what we're mapping back was bacteria. We have a very small amount of archaea here, a tiny amount of viruses, a little like unclassified, and then like some eukaryotic DNA. If we want to look more specifically at the bacteria, we can just click here on the kingdom bacteria. And it will give us a very dense thing here, but it will start to move and remove everything else and just kind of show what's in here. So if we want to get a better look, and we can click on something random, we can go to this genus up here. We can click here. It will load in. It will kind of show here on the right. We're at 8% of the total root, 8% of the bacteria, 22% of the phylum, 39% of this clade, 49% of this order, 95% of this family. And we can kind of come here and start to see like individual species that might be present. If we click on one of the species, we can kind of get an idea here, and we can start to look and see what's closely related, what percent of it made up like the total, what's the differences, layouts. I'll close that for the moment. And very often what can be useful here is if there's some kind of bacteria we're specifically interested in or something we want to particularly find, we can kind of investigate it. I'd encourage everyone, if you want to, mess around with this chart, especially if you pulled in the history, look around, see if you find any interesting bacteria. So you can click on here to like get back to the root here on the right. You can move around. Interesting things are, even with human reads removed, Kraken can be a little overly sensitive. And you can see, if we look here, we can find the mammals, and we can find primates, and we can say, hey, it's still saying there's a small amount of reads being mapped to humans, determined as by Kraken, even though we had tried to remove all the human read data from beforehand. So that's kind of our Chrono plot. I also want to take a moment here to show you things failing. Very often with how we've been hyping up Galaxy, it probably seems like everything works super smoothly and is always good, but inevitably when I was doing these analyses, I was doing things wrong, things happen, you error out, you hit out of memory. So that's kind of what 15 is here. So when something goes wrong in Galaxy, you get this beautiful red angry box inside your history to let you know something happened, and that you'll get a little thing that the error occurred here. When this happens, you can click this little bug report icon that will be new. Essentially, this will take you to this page here to help you interpret the error. We have an AI that will go through and try and convert the out-generated error into like a more human readable and understandable way, and possibly suggest ways you could maybe go about fixing it. I will say right now it's disabled. We're going to get used so much. We hit our credit usage limit, so we're swapping it out right now, but that should be back shortly. Additionally, there'll be troubleshooting steps and you can report a bug. So if you think there's like an actual problem, not with something you did, but something problematic with the tool itself, or something happened on the back end, like if a computer failed to initialize something or something went wrong, you can report it there. Additionally, the thing I wanted to show is this kind of data set details information page. This will be the little I link down here. And if you ever get an error or something goes wrong, this will have kind of like internal Galaxy information that's useful, but you might not need. It will tell you the input file you used and the parameters you selected. It will tell you the data set with the output that went wrong. And then it will also show you the command line that was executed. Very often, this is the same command line you can use with regular tools on a regular computer if you just installed the tool itself. The only thing is Galaxy will use like internal weird data names, so you just need to swap that to something more local. And then you'll also get kind of your standard output and standard error, which can be useful to look at. And then I'll also just point out, in addition to some of these, which can be very useful, especially if you're trying to troubleshoot or want to know what's really happening under the hood, you'll also get information here if you ever want to run it on your own of the exact amount of resources that were used for the job, how long it took, what it was assigned, and then kind of like the energy use. So although it'll be free, it'll give you an idea of like how much of environmental impact are you having, how much is it costing electricity, blah, 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 to use all of this, which can be useful. It's kind of just a nice, cool thing to learn. So with that, I figure we'll spend the last 10 minutes here on how to convert this all into a workflow and give it a run through. Very often, kind of what we're showing here is this is very much what you do with an exploratory analysis. Maybe you're going through a small subset of data, trying to figure out a workflow, make sure things work, or not a work, sorry, not trying to figure out a workflow, trying to figure out a process, make sure things work. But once you actually have it working, you're going to want to run it at scale, maybe on your 10 data sets, maybe on your full data set. You won't have to manually do it. And the best way to do that is through workflows within Galaxy. So not a history of this one. We'll still use the same history. I'll introduce you to how the workflow editor works, and then the easiest way to generate one really quickly out of our history directly without us even needing to do anything. So to get in the workflows, on the activity bar, you can just click workflow here. You'll have kind of a list of all the workflows you currently have available, and then there will be a list of all the public ones here that other people have made that you can access immediately. You also have the ability to import specific workflows, but for our purposes, we're just going to create a brand new one. This is going to take us to a little workflow editor like this, and we'll be able to build our own little workflow. So the first thing is oftentimes it's useful to have a name. So we'll just like, I don't know, we'll call it test demo. We can put an annotation if we want, and then we can also specify a license by default. So if other people want to use it or cite it or use it for their research, they have that ability. In terms of building out our workflow, much like our data, much like our regular Galaxy, the main thing, first thing we want to do is get our data in. So we can click on inputs here, and this will take in any kind of input we want. So for our case, we've only been working with a single data set, so we can select input data set here, and then we'll kind of drop it here on this grid for us. Once we create it here on the grid, it will open up on this right panel with information about it. So we can adjust the labels. Rather than input data set, we can say like subsample or whatever you want to call it, whatever name you want. We can specify if it's required for the workflow, if people need it. We can specify, hey, it has to be this data type, so people can't submit the wrong data type. If you leave this blank, it'll try and auto guess it. But will kind of give us this little spot to start with our data. And we can add as many of these as we want all in a row, move them about, whatever we'd like. After we have our first data set, the first thing we did was obviously run through FastQC. So we can search in here, On our inputs, just tools, kind of like the regular tool search, and we can click FastQC. And we'll generate FastQC and kind of stick it here on this grid structure as well. If we click FastQC, it'll open here on the right, and this will be the same tool form you would see normally in Galaxy. You can kind of expand it more if you want to have it be easy to read. It'll be the exact same options, exact same everything you'd see within Galaxy. You with the same help section at the bottom. The only difference is we can add a label if we want. So we can call this like quality control or whatever we want to specify it as for our purposes. The important thing for what we want to do with this is we need some way for our FastQC to know where to get its data from. And that's as simple as we can just grab the little node here at the end of our input data, drag it into the raw reads here, and now it's connected and we have our base, like most simple workflow. We have our data, our input data, FastQC, and we can just come over here and we could save it and give it a run, which we will do here in 1 sec. First, I want to point out there's a lot more options and a lot more things, and there's a lot of really good in-depth tutorials for fully building them out and making them nice. But there's always a best practices tab, and this will tell you things you can do to improve your workflow to make it look nicer and be more understandable to other people. In our case, we're missing some annotations to explain the steps. We didn't say who made it yet, and we have no labeled outputs yet, which we want to do. But for the moment, we can just give it a save. So we can just hit save current changes, and this will save our workflow. Once our workflow is saved, we can do something as simple as come here, run workflow. This will take us out of that editor and into this menu here. This is kind of the simplified form and how we'd recommend looking at this. Essentially, all that this needs is this is that input we put in called subsample. So we just need to pass in one kind of FastQ subsampled file. We can pass in our subsample DNA here that we pulled in. We can just hit run workflow. Upon hitting run workflow, this will take us to what it looks like when it's being run. So the very first thing it's going to do is it's going to initialize, figure out all the steps, make sure the data is in the right spot, and it will kind of take us to our graph. And here in a moment, what you should see is it will turn green. Now the subsample is loaded in. It's scheduling the job, and you'll see quality control turn yellow and then green as it finishes, and puts it into our history up here. So these will be the future FastQC outputs once it finishes running in queues. Additionally, you can view it through all the steps. In our case, the one we just made is super, super simple. It's, 2 steps, not much going on. You can see our inputs are loaded in. And then we'll also get real-time metrics as the job actually starts running for how long it's taking to run, aggregated times. And then we'll show statuses of things that are working. In the interest of time, I will skip ahead and ignore this part and instead show, since I killed those jobs, it's going to fail because I just canceled them in there. But I will instead show maybe the better way to generate a workflow rather than having to do it by hand, especially if you can do an exploratory analysis. In any history, you have the option to come up to history options. You can extract a workflow from it. And essentially what this will do is we can name it. So we call it like the NCAT workflow. And this will show every single step we've done and all the history items that we've created. And we can check which ones we would want to include. So we want the data to come in. We're going to want FastQC, fly, bandage. We probably want minimap. And then maybe we want the coronas. And I guess in this case, Kraken would usually populate, but I had pulled it from a different history. So it's not finding exactly where that's from. It wasn't done live in this history. But we can pull it all there and then create the workflow. And then we can come look at our workflows. Actually die. Let me create that correctly. That. And you can see here, we kind of created that workflow where it auto-populated in almost everything we need. And then the nice thing is here, we just click auto layout and it will move everything around. It'll pull in all the tools. If I hadn't broken the history a little bit, it would auto have all the connections too, but maybe the best example is here. You can see the history connected into here. Just reconnect it to Fly, and it populates. So that's an easy way to get all of the tools you used immediately into a workflow, and at least some of the starting connections. And with that, I also wanted to spend maybe the last 4 minutes before we do questions and things for as long as you guys would like on the IWC. Very often, the best way to get a good idea of workflows is to use ones that are like highly vetted, curated, and tested. And so one of the new things that we've been adding is we've had the IWC workflows for a little while. There's a whole repository, but we've been implementing a much nicer site to be able to access them, explore them, and find them. And so I want to spend like 1 minute kind of showing it off. And here is about 200 pretty documented workflows. They're kind of organized by different categories. If you click one of the categories, you get a bit more information about it, and then all the individual workflows. So for example, these are all the VGP workflows that are used for all those reference genome assemblies that are being generated. If, say, we are interested in the scaffolding with high C data one, all that we need to do is click it, and it's going to kind of load in, tell you what your inputs and outputs are, give you information about the workflow. It will also give us a diagram of what it actually looks like within Galaxy, so what the workflow looks like, all the different tools. We'll have access to like kind of a version history. But the most useful thing is we can specify which Galaxy instance we want it to go to. So in our case, use Galaxy. We can just click Run Workflow. And it'll import this entire workflow directly into our Galaxy and give us full access to it. So this will be the run form. And if we want to edit it, it will give us access to the entire workflow already pre-done in the editor. So if we want to modify anything or make any few changes, we have that option too. The other option is on this website, not currently, but shortly, they'll also have functional example data. So if you just click this Run with Example Data, it will import it into Galaxy with small test data that will work, and you can immediately run the workflow and inspect all the outputs and look through and see what it looks like. I'd highly recommend checking that out when it's ready. All right, so with that, I'll just finish up briefly and then take any questions. I just want to do one more call out. This was very brief, kind of high overview level. I want to point out the other training resources. The training, Galaxy training page itself is truly incredibly useful. No matter, it covers so many different analyses and in depth on the science and why you do things, as well as how to do it in Galaxy. But even if you don't want to do it in Galaxy, it's still very useful for having an idea of what tools are run, why you use them, and kind of like teach you how to do that. I'll also point out the Training Academy if people want to come. That'll be way more in-depth tutorials. Most will be online. There'll be constant support. If you want to do single-cell RNA-seq or spatial omics or something, that's a good spot to do that. There'll be tons of people doing it. It'll be kind of cool. And then finally, I just wanted to give a couple quick thank yous to the US Galaxy team for everyone who's working on making this all function, from all the compute resources to all the computer development, to the tool wrapping, to the integration. A thank you to the broader community as a whole. And then a couple more acknowledgements to everyone who's been helping out. A quick shout out to the GTX Foundation, which is in honor of one of the founders of Galaxy Project who passed away, that helps fund students to go to conferences and other charitable endeavors. And then let you guys know that if you have any questions, these are all of the links you can contact us at. People respond pretty quickly on Element, especially. That's usually where all the developers hang out who work on the project. And thank you, and ask if anyone has any other questions.
If there aren't any other questions, we'll stay on for just a little bit just to make sure. But I just want to say thank you so much to Dr. Schultz and Dr. Collins for your great presentation today. I am not a Galaxy user, but now I feel a little bit more comfortable about using Galaxy. So thank you so much. Is there any more questions before we end the workshop for today? I will also be sending out the recording for today and also the slides that they have provided. And again, I will be also sending out the post survey as well. So few minutes if anyone has any other questions. I think there's a question in the chat. And you can come off of mute too as well if you need to do that as well.
So I will say in addition to the workflows, you can run multiple samples through Galaxy pretty easily. We didn't go into it, but there's like kind of a system called collections, which if you can think about it as like a folder with multiple files in it, where you can run the same thing over and over again. So that's one way to do multiple samples. So like in those tool forms, you can say, run this on all 15 of my samples without it being a workflow. And in terms of taxonomic comparison analyses, there are different ways you can do that in Galaxy as well. I don't want to say, depending on which one you want exactly, I don't know them all. There's tons and tons of them, but that is also possible.
Any other questions? Dr. Graves, did you want to say something as well?
I just wanted to thank you guys again for a really well-constructed workshop. To say that my mind was blown would be an understatement. The worst thing you could do is give a tool like this to someone like me, an evil genius.
All right, well, if there is no one that has any other questions, thank you again so much for joining the GRADS-4C Galaxy workshop. Please be on the lookout for more workshops or seminars. You can always find us on our GRADS-4C communications tool, our LinkedIn profile, and just from word of mouth. So thank you again so much for this presentation. It was actually really, really great. Thank you.
Thanks, everyone. Thank you so much.
Much.
Guys.
Yeah.